All right, so I want to welcome everyone back. This is going to be our screencast for Chapter 7. And if you remember back to Chapter 6, we had talked about primarily the eukaryotic cell. And when we discussed the eukaryotic cell, we had talked about the different cell parts. What we're going to do in 7 is we are going to focus primarily on the cell membrane. And we're going to look a little bit deeper into the structure and see how that cell membrane is put together. And we're also going to look really heavily at what that cell membrane does for the cell. Now, when you think about the cell membrane, oftentimes we are going to compare that cell membrane to something called a fluid mosaic. And we call it a mosaic because it actually has lots of different other types of materials that are going to be embedded within that phospholipid bilayer that we had talked about back in Chapter 6. Now, the plasma membrane is considered the boundary that's going to separate the living cell from its surroundings. We consider the cell membrane selectively permeable, and what that implies is that it is only going to allow some substances to actually cross it more easily than others. So it's going to be very selective in what it lets in and what it actually lets out. The phospholipids, of course, are going to be considered the most abundant lipid in the plasma membrane, but as we kind of get further on in the screencast, we are going to talk about some other types of lipids that are going to be found there. The phospholipids are considered amphipathic molecules, and what that means is that they actually contain hydrophobic, which is those water-hating regions, and they do have hydrophilic regions, which are the water-loving regions. Now, the fluid mosaic model is going to state that a membrane is a fluid structure, so it's going to have a little bit of movement to it. And again, it is a mosaic, so it's going to have different types of proteins, carbs, and other types of lipids that are going to be embedded within it. Now, when you look at the different models of the cell membrane, the membrane has been chemically analyzed, and it's been found to be made mostly of proteins and lipids. Scientists studying the plasma membrane reasoned that it must be a phospholipid bilayer. A person that I would like you to note is somebody by the name of J. Singer and another person by the name of G. Nicholson back in 1972. And those two were actually the ones that proposed that the membrane is a mosaic of proteins sort of dispersed within the bilayer, with only the hydrophilic regions being exposed to water. And so again, if you look down here towards the bottom, you can see the phospholipid bilayer right here. And this is what you had seen back in Chapter 6. I think we had even talked about the different proteins that would be embedded within the layer. And if you notice, even these proteins are going to have hydrophobic regions and hydrophilic regions, very similar to what you would see within this phospholipid bilayer. So the head region of the bilayer is going to be considered the hydrophilic head, so that's going to be the water-loving head of the layer. And then the hydrophobic regions are going to be sort of in between those hydrophilic heads. So it's a bilayer, so there are two layers there, but it's going to be kind of trapped in between those two hydrophilic regions. Now again, the phospholipid bilayer, the plasma membrane in general, is considered to be very fluid. It says that the phospholipids in the membrane can actually move within the bilayer. Most of the lipids and some of the proteins will actually drift laterally, so this is going to mean that they're going to move this way and this way within that layer. Very rarely is the molecule going to flip-flop transversely across the membrane. So in other words, sort of turn itself upside down. As temperatures cool, so temperature is going to have a big play in this, membranes are actually going to switch from a sort of fluid state to a more solid state. And if you think about water, as water cools, it does become more solid. The temperature at which a membrane solidifies is going to depend on the different types of lipids that you would find in that membrane. So membranes rich in unsaturated fatty acids are considered more fluid than those rich in saturated fatty acids. Now, if you remember back to our discussion about the macromolecules, unsaturated fatty acids were a large component of oils. And so it kind of makes sense that those would be more fluid. And those that were considered saturated fatty acids were going to be the more solid type of fats, like butter and lard. Membranes must be fluid to work properly. So again, if we make a comparison between this membrane and its fluidity, sometimes they are compared to salad oil. So that kind of gives you an idea as how fluid they are. So again, as we had said, there's going to be lateral movement between these different molecules within this bilayer. Again, the flip-flopping of those molecules doesn't happen too often. It says here about once per month. The down here towards the bottom, again, if you look at membrane fluidity, again, you can see these unsaturated hydrocarbons, and you can see the kinks that you would see in the fatty acids that kind of indicates those double bonds. Um, this kind of shows you how, again, it's more fluid within this type of hydrocarbon, again, these unsaturated fatty acids, as opposed to the saturated ones, which are a little bit more solid. And as I had said, sometimes you will find other types of fats or lipids within this bilayer. And so down here you notice we have a good example of that, which is going to be this cholesterol, which is embedded within that layer. So the steroid cholesterol that we saw in the previous slide, again, it's going to have different effects on membrane fluidity at different temperature. At warm temperatures, such as around 37 degrees Celsius, cholesterol is actually going to restrain the movement of those phospholipids. But if you cool it down a bit, it does actually maintain the fluidity by sort of preventing that very tight packing that you would see if it was primarily just that phospholipid bilayer. So again, it kind of prevents that kind of compact nature of the membrane. So the membrane proteins, again, are a very important part of the cell membrane. So a membrane is a collage, which basically means a very large variety of different proteins sort of embedded within the fluid matrix of the lipid bilayer. And you can see that being represented right down here. Proteins are going to determine most of the membrane-specific functions, in other words, what it's going to do. We do have different types of proteins. One of them is called a peripheral type of protein, and these are going to be bound to the surface of the membrane. We have another one called an integral protein, and these are going to penetrate the hydrophobic, again, that water-hating core of that phospholipid bilayer. And again, those integral proteins that span the membrane, so this is just going to be a different variety of this protein, are going to be called transmembrane proteins. And so again, down here towards the bottom, you can see a good representation of the cell membrane. You can see that ECM that we had talked about back in Chapter 6, that extracellular matrix right above the membrane. You can see some representations of some glycoproteins. And again, as I had mentioned, there are some carbs embedded there as well, some glycolipids over here. And again, you can see down here, this would be considered an integral protein. This would be considered a peripheral protein. And again, you can see these cholesterols, and then the cytoskeleton that you see right beneath that phospholipid bilayer or that cell membrane. So there's going to be 6 major functions of these different membrane proteins. One of them is going to be transport. The second would be enzymatic activity, so the ability to sort of speed up chemical reactions within the cell. A third is going to be signal transduction. The fourth is going to be cell-to-cell recognition, so there's going to be some communication going on between cells using the cell membrane. The fifth is going to be an intercellular joining. And then the sixth is going to be attachment to the cytoskeleton, and again, working within that extracellular matrix or ECM of the cell. So down here, or actually over to the right, you can see the diagram that sort of represents all 6 of these. Here you can see the transport mechanism being represented, in other words, the transport of maybe molecules back and forth across the cell membrane. Some of that does require energy, so in the form of ATP. Enzymatic activity, again, trying to sort of promote a really fast occurrence of different types of chemical reactions. Signal transduction, again, kind of communicating different types of messages across that cell membrane. Cell-to-cell recognition, so if you have 2 cells that get really close to each other, they sort of recognize each other with these different types of proteins, these glycoproteins. Intercellular joining, so sort of a mixing, not necessarily a mixing, more of a fusion between the cells. And then, of course, attachment to the cytoskeleton, and again, the extracellular matrix. So making sure that that stays very close to the cell membrane. So we have proteins that actually help in that. So the role of the membrane carbohydrates is primarily in the cell-to-cell recognition function of that membrane. So cells are going to recognize each other by binding to surface molecules, often, again, as we had mentioned at the very beginning of the screencast, carbohydrates on the plasma membrane. Now, membrane carbohydrates may be covalently bonded to the lipids, forming glycolipids. And again, remember, covalently bonded means the sharing of those electrons between those molecules or elements. Or more commonly to proteins, which are going to form what we call glycoproteins. So whenever you see the word glyco in front of these terms, again, it sort of implies an interaction between carbohydrates and that other molecule, whether it be lipid or proteins. Carbohydrates on the external side of the plasma membrane are going to vary among species, individuals, and even the cell types that you would find within an individual. So again, the carbohydrates are very important when it comes down to this whole communicating factor of cell membranes. So membrane structure is going to result, as we had said earlier, in selective permeability of that membrane. So a cell must exchange materials with its surroundings, which is going to be controlled by that plasma membrane. So they're going to be considered selectively permeable, basically regulating the cell's molecular traffic. Now the hydrophobic or nonpolar molecules, for example, such as hydrocarbons, can actually dissolve in the lipid bilayer and actually pass through the membrane pretty easily, in other words, very rapidly. The polar molecules, on the other hand, for example, such as sugars, don't have it quite as easy, and so they need a little bit of help when it comes down to getting across the membrane. So we do have special proteins called transport proteins, which are going to allow the passage of the hydrophilic substances across the membrane. Some transport proteins called channel proteins are going to have a hydrophilic channel that certain molecules or ions could actually use as a tunnel. And in fact, when it comes down to the passage of water, we have a special channel protein called an aquaporin, which is going to facilitate or help the passing of that water across the membrane. Other transport proteins called carrier proteins are going to bind two molecules and actually change the shape to shuttle them across the membrane. And so a transport protein is, again, going to be very specific for the substance that it does tend to move. So the shape is going to be specific to that type of molecule that is trying to get across the membrane. Now, when you talk about the cell membrane, we need to talk about different types of transport of those molecules across the membrane. The first one we look at is something that is called a passive type of system or a passive type of transport. So what we're going to be doing is we're going to be transporting materials across the membrane, but there is going to be absolutely no energy investment in this process. So diffusion is going to be a tendency for molecules to spread out relatively evenly into the available space. If you think about perfume, for example, if you have somebody that sprays perfume, initially all of that perfume is going to be pretty much concentrated around that one individual, but eventually over a period of time, that perfume is going to permeate throughout the entire room, and you're going to be able to smell it, even if you're not next to that person who just sprayed it. Although each molecule is going to move randomly, diffusion of a population of molecules may exhibit what we consider a net movement in one direction. So at dynamic equilibrium, as many molecules cross one way as cross to the other direction, and that's what dynamic equilibrium is going to refer to. So in other words, going back and forth until you have approximately the same concentration of molecules on each side. The movement never stops, but you have about the same number moving this way that you would have moving this way. Now, substances are going to diffuse down their concentration gradient, and that gradient is going to be the difference in concentration of a substance from one area to another. Now, no work must be done to move substances down the concentration gradient. Again, when you think about work, you think about energy. And when you talk about passive transport, again, there's no energy input by the cell. The diffusion of a substance across a biological membrane, again, as we had said, is considered a passive type of process because, like I said, it requires no energy from the cell to actually make it happen. So over here on the right, this is just a really quick example of diffusion. And again, when we talk about net diffusion, we're talking about the molecules that you would see right here. This would represent the membrane that you see in this particular example. And because of the random motion that is occurring here, eventually those molecules are going to make their way through the openings in this particular membrane. And eventually, after a period of time, you're going to notice that we're going to have approximately the same number on each side. And if you notice over here, when you think about two different types of molecules, maybe a different one on each side, then the same thing happens. If they're more concentrated on one side, after a period of time, the purple ones that you see will make their way to the left, and the orange ones that you see will eventually make their way to the right until we reach equilibrium. Now what we want to do now is we want to talk about a special type of diffusion, and this type of diffusion is called osmosis. Now osmosis is the diffusion of water across a selectively permeable membrane. Remember, selective basically means the membrane can determine what goes across and what doesn't. Water is going to diffuse across a membrane from a region of lower solute concentration to the region of higher solute concentration. So one thing to note here, we're talking about the movement of water in this case, not the movement of the solute or the other molecules you might find within that water. Now over here on the right, this is considered the YouTube experiment, and what you notice about it is we have water on both sides, but we have a different concentration of sugar on each side. There's more on the right than there is on the left. And what you'll notice after a period of time is eventually the water on this side of the tube is going to get higher. Now the reason for that is because the amount of water that you would find on this side of the tube is going to be less than you would find on this side. So there's going to be a natural tendency for the water to move to the right. Again, remember we go down that concentration gradient from high to low. And because that water is moving to the right, it's going to appear that the level in this tube is going to rise up. Now essentially, if you think about it, these molecules in green, this sugar, can't pass through the membrane. So even though it looks unevenly distributed, when you think about the ratio of the glucose or the sugar to the water, they actually are in a state of equilibrium. The ratio of the two are actually the same. So down here towards the bottom, again, this represents the selectively permeable membrane, which basically is telling you the water can get across, but again, the glucose cannot. Now when you talk about osmosis, sometimes we refer to something called the water balance of the cells. And we do this in actually a couple of different ways. The first way we're going to look at is actually the water balance of cells without cell walls. And sometimes you'll see this word over here is called tonicity, and this is the ability of the solution to actually cause a cell to either gain or to lose water. Now these are three terms you probably heard back in honors biology or general biology. One of them is isotonic, one is hypertonic, and the third is hypotonic. And all three of these refer to the solution that that cell is actually placed in. Now when you talk about an isotonic solution, the solute concentration in that solution is going to be the same as that inside of the cell. Now because of that, there's going to be really no net movement of water across the plasma membrane. Now I'm not saying the water isn't moving back and forth, I'm saying we don't have more water going to the right as opposed to the left or the other way around. Water is still passing back and forth, but it's at about the same rate. Now if you put an animal cell in a hypertonic solution, the solute is going to be greater than the inside of the cell. And so what's going to happen is that the cell itself is actually going to lose water. So if you think back to our YouTube experiment in the previous slide, hopefully that should make sense because again, you think about it, in a ratio sort of way, there's actually more water inside of the cell than there is on the outside because the solute is greater on the outside. And so the cell is going to tend to lose the water. The water is going to rush out of the cell, and that's going to cause the cell to shrivel up. Now if you take that cell and you put it into a hypotonic type of solution, in this case the solute concentration is actually less on the outside than you would find on the inside of the cell, and because of that, what's going to happen is the cell is actually going to gain water. Because remember, there's more solute on the inside in a ratio sense than there is on the outside, and so that water is going to tend to rush in. So over here on the right, you can see this being represented. Again, we have a hypotonic, isotonic, and hypertonic solution. And so in a hypotonic situation, remember that water is going to rush in. And when it rushes in, unfortunately for animal cells, there's no cell wall, and that cell tends to lyse or to burst. In an isotonic type of solution, again, remember the cell is not going to really change too much. The amount of water going in is going to be the same as the amount of water going out. And in a hypertonic type of solution, remember the cell is going to shrivel up in animal cells because again, that water is going to tend to rush out of the cell. Now if you look down here towards the bottom, we have a plant cell being represented as well. And pretty much the same thing happens, but the only difference is that because there is a cell wall, we do not see the lysis actually happening in a plant cell because the pressure can be contained with that cell wall. And we're going to talk more about the plant cell in the next slide. So actually, we're not going to talk about the plant cell quite yet. We're still going to still focus a little bit more on the animal cell. So when you think about the hypertonic or hypotonic environments that are created in some cells, the unfortunate thing is for a lot of single-celled organisms, this can present a problem. And so what has happened is they've actually evolved or developed ways to sort of fix this. So they are able to maintain osmoregulation, and what that is basically the control of the water balance within their cell. And it's really important when it comes down to life in general. Now, a good example of this would be the paramecium. And so if this paramecium, which again is hypertonic to its pond water environment, if you place that within the environment, it actually has a contractile vacuole that's going to act as a pump. Now what that means is this structure right here, it's a vacuole, it's just a sac that you would find within the cell. As that sac fills up with water, it actually has the ability to squeeze or contract that vacuole, and so that's going to move the water out of the cell. So that's a way for it to sort of maintain the proper level of fluid or water within the cell as compared to the outside of the cell. Right, so what we're going to do next is we're going to look at the water balance of cells with cell walls. So we're no longer talking about animal cells, now we're going to talk about plant cells. So the cell wall is actually going to help maintain water balance inside of a cell. So a plant cell in a hypotonic type of solution is going to swell very much like you had seen in the animal cell. Only this time the wall is going to oppose the uptake of that fluid or water. And the cell is actually going to become very turgid or very firm. Now, if a plant cell and its surroundings are isotonic, then there is no net movement of water into the cell. And because of that, the cell is actually going to become very limp. And the plant is going to appear as if it's wilting. Now, in a hypertonic type of environment, the plant is going to lose water. So those cells are going to lose water. Eventually, the membrane itself is going to pull away from the cell, and unfortunately, this is usually lethal for most plant cells, and we do call it plasmolysis. And what you see over here on the right is a good example of plasmolysis. So if you notice, these are, I believe, onion cells, and they've been stained, and so this area right through here is going to be that central vacuole. Now, if you notice, it's filled with water, but if you subject it to a hypertonic type of environment, then what's going to happen is all of that water is going to be drawn out of the cell, and again, it's going to cause that membrane to pull away from the cell wall. So the next thing we need to look at is something called facilitated diffusion. And to facilitate something means to help something out. And so this is going to be a form of passive transport that's actually going to be aided or helped out by special proteins. Now, in facilitated diffusion, transport proteins are going to speed up the passive movement of molecules across the plasma membrane. So these are going to be molecules that can't normally pass across that phospholipid bilayer, but they need a little bit of help to get across. So channel proteins are going to provide corridors that allow a specific molecule or ion to cross the membrane. So they are very specific to certain molecules. And so there's lots of different types of channel proteins that are going to be embedded within that membrane. Now, channel proteins include aquaporins, and these are going to be used to facilitate the diffusion of water. And we also have those that are called ion channels, and these are going to be the ones that are going to either open or close in response to some sort of stimulus. And they're sometimes called gated channels. Now, carrier proteins are going to undergo a subtle change in shape that's going to translocate the solute binding site across the membrane. So what that really means is simply that the protein that you see over here is going to basically morph to the shape of the molecule that needs to be carried across, and then it's going to bind to the protein and eventually make its way to the inside of the cell. Now, as opposed to the passive transport that we had talked about in previous slides, now we're going to transition into a more active type of transport. Now, when you talk about active transport, you need to understand that the cell is going to need to provide a little bit of energy to move these solutes, because it's going to be moving these solutes against their concentration gradient. Now, what that means is instead of going from an area of high to low, now we're going to be going from an area of low to high. Now, facilitated diffusion is still considered a passive type of transport system, and that's the one we had talked about in the previous slide, simply because the solute is continuing to move down its concentration gradient. In other words, going from high to an area of low. Now, there are some transport proteins, however, that can actually move the solutes against their concentration gradients, in other words, going from low to high. Now, active transport, again, as we said, is going to move these substances across their gradient. Active transport is going to require energy, and most of the time that energy is going to be in the form of ATP. Active transport is going to be performed by specific proteins, again, embedded within the membrane. Now, active transport is going to allow cells to maintain a concentration gradient that is going to be a little bit different from their surroundings. Now, one special type of active transport system is called the sodium-potassium pump, and that's one we're going to look at in the next slide. So what we see here is a good example of the sodium-potassium pump. And so if you notice from this diagram on the left, this is going to be the inside of the cell, and this is going to be the outside of the cell. And if you notice, sodium is going to be in a relatively high concentration out here, and potassium is going to be in a relatively low concentration in this area of the cell on the outside. As opposed to the cytoplasm, where sodium is typically pretty low and potassium tends to be pretty high. And so what's trying to occur here is we're trying to make sure that we get as much sodium out of the cell as we possibly can. And so this purple blob that you see right here is going to be the protein. And so that's going to be the protein that's going to basically help get these sodium ions across the membrane. But again, because sodium is in such a high concentration on the outside, we're going to need to use a little bit of energy in the form of ATP to sort of push those sodium ions out of the cell. Now, as opposed to potassium, which is what you see right down here, we're trying to get as much potassium into the cell as we possibly can. But remember, potassium tends to be in a high concentration on the inside of the cell, but the cell tries to take in as much as it possibly can. So with a little bit of energy, we're going to end up taking that potassium and moving it into the interior of the cell. So again, this is a form of active transport, primarily, again, using that ATP supplied by the cell, and we're moving from an area of low to an area of high concentration. Now, this diagram is simply comparing the different types of transport that we've talked about up to this point. So again, remember, talking about diffusion, just a simple random motion of molecules kind of moving across that phospholipid bilayer, not really needing any help to get across. Facilitated diffusion employs the help of some proteins, and they're used to sort of kind of guide or encourage those molecules across, but again, without the need for energy. So it's still somewhat of a random type of situation because it's just by chance that these molecules hit the protein in the right spot and make their way across. But then when we talk about active transport, remember active does require the input of energy by the cell. And so we're trying to get molecules that typically tend to be in a higher concentration on the inside. We're trying to get even more of those molecules across that cell membrane. And so energy is necessary to, again, go from an area of low to an area of high that you see down here. And remember, these two go from an area of high to an area of low concentration. Now, every once in a while, you're going to run across a cell that's going to need to bulk transport different types of materials across the plasma membrane. And this is going to occur by either exocytosis or endocytosis. Exo is going to be out and endo is going to be in. Small molecules in water will enter or leave the cell, again, through the lipid bilayer or the transport proteins that we had talked about. But when you talk about larger molecules, such as a polysaccharide and some proteins that are pretty big out there, they're going to cross the membrane in bulk, and it's going to be via the vesicles that we had talked about back in Chapter 6. Now, bulk transport is going to require energy on the part of the cell, so this is a form of active transport. When you talk about exocytosis, you're talking about transport vesicles that are going to migrate to the membrane. They're going to fuse with that membrane, and then they're going to release their contents. Many secretory cells are going to use exocytosis to export their products. And a lot of times you'll have different types of hormones and various materials such as that are produced by the cells that are going to be exiting the cell in this bulk transport method. Now, in endocytosis, we're going to talk about the cell actually taking in macromolecules, so really big molecules, by forming vesicles from the plasma membrane. And endocytosis is simply the reverse of exocytosis, and again, involving different types of proteins. So there are actually three different types of endocytosis. The first one is phagocytosis or phagocytosis, and this is where a cell actually engulfs a particle in a vacuole or a vesicle. The vacuole or vesicle is going to fuse with a lysosome, and it's going to be used to digest the particle, and a lot of single-celled organisms are going to participate in this. Then there's pinocytosis, which involves molecules that are taken up when extracellular fluid, so fluid on the outside, is actually gulped into tiny vesicles formed by that membrane. And then we have something called receptor-mediated endocytosis. So these are going to be the binding of ligands to receptors that trigger the vesicles to form. Now, a ligand is any molecule that binds specifically to a receptor site of another molecule. So over here on the right, this is going to be considered phagocytosis, and so here you notice we have some pretty, what I consider chunky material on the outside of the cell, and so this is going to be found in the extracellular fluid on the outside, and if you notice we have a food vacuole that's going to form, and that food material is going to be taken into that food vacuole, and again, by actually combining with the lysosome, it's going to work to digest that food. Down here is pinocytosis, and so what we have is we have in this extracellular fluid a collection of molecules that are going to be sort of dumped within this vesicle and then of course carried to where it needs to go. Then down here we have receptor-mediated endocytosis, and so right here these are going to be considered the receptors. And these are going to be considered the ligands. And so when they actually meet up with these receptors, they tend to trigger the formation of that vesicle that you see right here. And so that's going to basically encourage the uptake of the molecules that are out here. And then of course you notice we have what we consider a coated vesicle here. So this vesicle is a little bit different than what you would see in the previous two. And it's all based on the, again, binding of that ligand to that receptor molecule on the cell membrane. All right, so that's going to finish up our Chapter 7 screencast. Please make sure that you have completed your Chapter 7 study guide.